Welcome everyone. Do you see my screen and do you see my mouse on the screen?
Yes, it's perfect.
Perfect. Okay, so thank you. Thanks for the invitation. It's always a pleasure. So I very much enjoy the conference. Great work, fancy tools. So this is really nice. So I'm going to talk about the role of serotonin in regulating ultrasonic vocalizations. I'm going to focus on pop ultrasonic vocalizations in the beginning, mice and rats, and then move on to different types of vocalizations emitted by rats. So serotonin is a very important neurotransmitter. It's involved in regulating many different psychological, but also physiological processes, ranging from thermoregulation, appetite, pain, to mood, social behavior, including aggression, for example, and mating behavior and things like that. But it also plays a very important role in neurodevelopment. And so there's the TPH2 deficient mice. So TPH2 is the rate-limiting enzyme and that is needed for serotonin synthesis in the brain. So those TPH2 animals are deficient for TPH2, they don't have serotonin in the brain. And as you can see here in the study published a while ago, as you see, the lack of serotonin in the brain leads to a massive effect in terms of growth retardation. So the TPH2 deficient animals lacking serotonin in the brain, specifically in the central nervous system specifically, they are much smaller. So there's no serotonin in the brain, and here you see the body weight gain. So you see eventually they're going to catch up, but there's a massive delay, a strong reduction in growth and body weight gain. And this is associated with a quite significant reduction in survival, as you can see here. About 40 to 50% of the TPH2 deficient animals lacking serotonin in the brain actually die. So we were wondering whether this is actually driven by the lack of serotonin, a direct effect of the lack of serotonin, or whether this is driven by some related factors, as for example, impaired communication between mother and pup. So we decided to focus on pop ultrasonic vocalizations because we thought, well, serotonin plays a very important role in social behavior and communication. So maybe the communication of the pups is impaired, leading then to reduction in maternal caregiving behavior. So that's why we focus on the pop ultrasonic vocalizations. We had a very nice talk about that yesterday already. And it was also mentioned in Bernard Engelit's presentation right before. So pop ultrasonic vocalizations, they were discovered around 70 years ago by Cepheus and Schleid in a number of different species shown here on this slide. Unfortunately, the study is not very well known, probably because back then it was still written in German. I think it's unfortunate because they came up already back then with two very important hypotheses that really still do have a very significant impact on the field. So they got inspired by also the work of Konrad Lorenz, and they came up with the two hypotheses saying that first, those calls actually do reflect an affective state, a negative affective state, because obviously situations that elicit those vocalizations, they are very dangerous for the pup. So when you separate the pup from the mother and the littermates, it's very dangerous. There's no protection, there's no food, no warmth, no shelter. So it's very dangerous situations. So those calls probably reflect a negative affective state, and they probably serve communicative functions as contact calls. So what Cepheus and Schleid observed is that mothers leave the nest in response to pups that are able to vocalize and search for the pups and retrieve the pups back to the nest, but only in case the pups are able to vocalize and not in response to pups unable to vocalize. So we then studied those vocalizations in the TPH2 deficient animals lacking serotonin in the brain. So we first confirmed the growth retardation phenotype. And as you see here, starting basically from day 3, 4, 5, there's a significant reduction in body weight. So at day 9, postnatal day 9, wild type and heterozygous animals, they weigh around 5 grams. But the mutant animals lacking serotonin in the brain, only about 2 grams. And not surprisingly, this is associated with a reduction in body temperature, impairments in body temperature regulation, also studying around day 3-4 during development. So we were wondering whether those deficits are actually driven by impaired communication between mother and pup. And indeed, there's evidence for that idea. So what we found is that the TPH2 deficient animals that lacking serotonin in the brain, that those animals actually produce way fewer ultrasonic vocalizations, particularly during the first week of life, suggesting that the reduction in pup ultrasonic calling leads to reduction in maternal caregiving behavior, reduction in feeding behavior, for example, which then results in the growth retardation phenotype. So this was done in mice, but we decided to see whether this is actually also true in rats. Recently, a study conducted by Tianhua Wang in our lab in combination with a potential rescue strategy. So we basically switched from mice to rats, but it's TPH2 deficient rats lacking serotonin in the brain. We again focus on the pup vocalizations. So in this study, we assessed pup ultrasonic vocalizations every second day during the first 2 weeks of life. As I said, we did not only switch species, but also actually we decided to apply a potential rescue strategy. So we compared standard nesting, so 1 mom with the litter versus 2 moms with the 2 litters, because it was shown repeatedly that communal nesting can improve a couple of social aspects related to social behavior, survival, growth, also cognitive functioning and things like that. So it's basically a 2x3 design as illustrated here. So wild-type heterozygous knockout, standard nesting versus communal nesting. So we then recorded those pup ultrasonic vocalizations. So you see a rat pup emitting those pup calls, and while showing circling behavior, making sure those calls are emitted in all the different directions to increase the chance to be localized by the mother and retrieved back to the nest, actually. So what we find is that the phenotype is first observed in mice, actually also true and visible in rats. So we see a significant reduction in pup ultrasonic vocalizations. This is reflected in call numbers, but also total calling time. And as you see here, this is still seen after communal nesting. So communal nesting does not rescue this phenotype. There's still a significant reduction in pup ultrasonic calling. And similar to the mice, this starts to emerge around postnatal day 2, so a bit earlier than the growth retardation phenotype, suggesting that this is really driving the growth retardation phenotype, at least partially. In terms of the acoustic features, so what we find quite interesting is we do see in pup ultrasonic vocalizations, in rats, we do see 4 clusters basically. In terms of frequency, a lower frequency cluster around 40 to 50 kilohertz, low frequency modulation or high frequency modulation, and then another one between 60 and 70, a low frequency modulation and high frequency modulation. So 1, 2, 3, 4 clusters. What we find quite interesting is that in the knockout in the animals lacking serotonin brain, that this 4th cluster under standard nesting as well as communal nesting conditions, that this 4th cluster is completely lacking basically. So the most complex call type is not really present in the mutant animals lacking serotonin in the brain. The idea is that those calls induce maternal caregiving behavior such as retrieval behavior as shown in this video here. So we basically ask whether the difference in pup ultrasonic calling, whether this actually leads to affects maternal preference. So we did a maternal preference assay. So the mother can choose whether she wants to spend time with the knockout or the wild type, the knockout producing fewer calls than the wild types. And overall, we see that the mothers show a clear preference for the wild type animals, supporting the idea that the impairment in pup ultrasonic calling indeed leads to a reduction in maternal preference, leads to a reduction in maternal caregiving behavior, which then results in a growth retardation phenotype. Interestingly, this preference of the mother is most prominent under communal nesting conditions. So in this more competitive situation, there's a clear preference for the mother for the wild type conditions, for the wild type pups over the knockout pups. Another interesting phenotype that was associated with the lack of serotonin is maternal affiliation, maternal affiliation deficits. So this is a study where they studied mice, rats, and monkeys lacking serotonin in the brain. And what is found is a reduction in maternal preference. So in mice and rats, we use this homing test. So they can basically decide whether they want to stay in the center, or whether they're going to crawl into the area with the soil bedding containing the odor of the mother, or in another compartment containing clean bedding. And wild-type animals and heterozygous animals, they show a clear preference. Mice as well as rats show a clear preference for the maternal odor, show intact maternal affiliation, whereas there's no preference whatsoever in the TPH2 deficient animals lacking serotonin in the brain, in mice as well as rats. And actually, in a similar assay, this is also true for monkeys. So we were able to replicate this phenotype. So here you see a very nice preference of the wild-type and heterozygous animals for the odor of the mother, intact maternal affiliation behavior, and this was not present whatsoever in the knockout animals lacking serotonin in the brain. Interestingly, we were able to rescue this phenotype through communal nesting. So communal nesting can have positive effects. It just does not rescue the communication deficit, but it does rescue the maternal affiliation deficit. But it also has negative consequences in terms of body weight gain, for example, in terms of somatosensory reflexes. And I just want to highlight one here. Here, this line is the wild type, 100%. These are the heterozygous animals. They are about the same in terms of body weight as the wild type animals. But here you see the mutant, the knockout animals. And here you see they are much smaller than the wild type animals. Already under standard nesting conditions, but it even gets worse under communal nesting conditions, probably because there's more pups and it's even more competitive. And so this basically means certain features can be rescued, like maternal affiliations, others are not affected, like the vocalizations. And in such a competitive environment as communal nesting, there's an even stronger growth retardation phenotype present. So rats do not only emit those pup calls, but actually do emit many other call types, as you all know. So there's, for instance, the aversive 22 kHz calls and the positive 50 kHz calls. And I just want to briefly mention those two call types as well, because we studied phenotypes related to them also in the TPH2 animals in adulthood, then in response to psychostimulants. So aversive 22 kHz calls, they occur in aversive situations such as predator exposure or fear learning, for example. There's this very famous study by the Blanchards where they put those animals in a visible burrow system. So they were able to freely explore this environment. So the rats were living there for a couple of days, and then they put a cat into this environment. And what they observed is that, of course, they were hiding, they were running away from the cat, and that this was actually associated with the emission of those aversive 22 kHz ultrasonic vocalizations. So I still find this a very fascinating study, but of course it's difficult to work with cats in a lab. So that's why we were actually quite happy to see that this actually works also in response to urine samples of predators. So we did a study together with our collaborator Markus Wöhr in Magdeburg, where we exposed rats to predator urine, and in particular lion urine seems to be very efficient in eliciting those aversive 22 kHz ultrasonic vocalizations, which means you don't have to bring the lion to the lab. It's sufficient to bring the lion urine to the lab. So this is just one brief example about the 22 kHz calls. But as you know, there's also 50 kHz calls. They occur in positive situations. For instance, in response to play behavior or when the animal is tickled. So that's the famous work by Jaak Panksepp and Jeffrey Burgdorf and Brian Knudsen. And this is actually working very well during social interaction behavior. Juvenile animals playing with each other, they produce these 50 kHz calls. Besides natural conditions such as play behavior, or when this is mimicked, this play behavior through tickling, drugs of abuse, particularly psychostimulants, are very efficient in eliciting those positive 50 kHz calls. And this is a work done by Maria Willardson in the lab of Paul Clark. What we found there is that psychostimulants such as caffeine and amphetamine can induce a massive amount of those positive 50 kHz calls. This is actually dose dependent, as you see here. Increasing doses of amphetamine lead to increasing numbers of those positive 50 kilo scores. If you dose too high, the animals start to show stereotypies, and then they stop vocalizing. But moderate doses of 1 milligram per kilogram, 2.5 milligram per kilogram lead to a massive increase in 50 kilo calling. This is dependent on dopamine signaling, so you can block that by administering dopamine antagonists. But particularly interesting in the present context is that this is also dependent on serotonin. So this is dependent on the serotonin 5-HT2C receptor, as you can see here. So we were able to completely block amphetamine-induced 50 kHz calling. So there is an interplay between dopamine and serotonin in regulating those 50 kHz calls. We were recently suggesting that we can actually use those 50 kHz calls as a measure of mania. So mania, important aspect of bipolar disorder, characterized by elevated drive and elevated mood. So we're arguing that we can actually study both components in rats. So elevated drive by assessing amphetamine-induced increase in locomotor activity, and elevated mood by assessing amphetamine-induced 50 kHz calling. Because both of them can be blocked by lithium treatment, which is the gold standard to treat bipolar disorder, mania in the clinic. So this can be brought back to baseline for both elevated drive and elevated mood when administering lithium. So the idea is basically that under normal conditions, appetitive conditions such as play, they emit 50 kHz calls, but under conditions like amphetamine treatment, they show an excessive level of positive affective state. This is then associated with an excessive level of 50 kHz calls, and that this can be blocked by lithium treatment, which basically means that we can study this rather unique feature of mania in rodents. So what about the TPH2 animals? So the TPH2 animals, we obtained evidence that lack of serotonin in the brain actually affects the emission of 22 kHz calls and 50 kHz calls. I'm not able to show all the details. I just want to show one study where we assessed how TPH2 animals actually respond to MDMA. So MDMA is typically believed to not elicit, it's also a psychostimulant, but to not elicit 50 kHz calls. So basically what we did is we exposed the wild-type animals, the heterozygous animals, and the knockout animals first just to an open field. This first day, second day is then saline treatment, third day is MDMA treatment, and assess their behavioral response, including the 50 kHz calls. So what we found is that we do see actually quite a significant number of 50 kHz calls in response to MDMA. It depends a little bit on whether we compare that to baseline or to saline. We believe that comparison to saline is probably the more appropriate comparison because, yeah, during saline injection, they get an injection. During MDMA injection, they get an injection. And under those two conditions, the environment was not familiar anymore, whereas during baseline testing, they get habituated to the environment in the first place. And we know that novelty exposure can trigger 50 kHz calls as well. So when comparing to saline, we actually do see a slight increase in 50 kHz calling in response to MDMA, at least in wild-type animals. Of course, this is not as strong as in response to amphetamine, but still a significant increase, particularly in females, not so much in males. This effect is not there in the TPH2 deficient animals. So TPH2 deficient animals do not produce 50 kHz calls. There's no increase in 50 kHz calling in animals lacking serotonin in the brain. In terms of the acoustic features, we also do see changes in terms of call duration, for example, and we do see changes in the call clustering, as shown here. What's interesting is that the MDMA treatment seems to affect the call clustering. And what's particularly relevant, I think, is that at least in the TPH2 deficient animals lacking serotonin in the brain, we also do see 22 kHz calls. Quite a significant number and only a few animals, though. So it seems like whereas wild-type animals respond by producing 50 kHz calls, possibly reflecting a positive affective state associated with the MDMA, this is not the case in the TPH2 animals lacking serotonin in the brain. They do emit aversive 22 kHz calls, at least some of them, in response to this psychostimulant. What about behavior? in line with the vocalization data, but more robust, I would say. We see in wild-type animals in response to MDMA as a psychostimulant, a massive increase in locomotor activity. And this increase is basically absent in animals lacking serotonin in the brain. This is true for the animals' pooled data, males and females, but also when analyzing them separately, females versus male only, we see that there's basically no change in locomotor activity whatsoever in response to MDMA in TPH2 deficient animals. So under baseline conditions, we don't see much differences in the terms of the time course. This is also true in response to saline, even though the TPH2 animals tend to be a bit less active there as well. But in response to MDMA, the picture is very clear. So we see very high levels of locomotor activity across 45 minutes of testing, much higher than under saline conditions, obviously in response to MDMA. But this is not the case for the TPH2 deficient animals lacking serotonin brain. This is the red line here. So they basically don't move much. So they basically behave as if they did not receive the MDMA. With that, I want to conclude. So as you all know, rodents emit different types of vocalizations. They seem to reflect different affective states. They do have very important communicative functions. And if those communicative functions are impaired, for instance, in TPH2 deficient pups, this can have severe negative consequences, such as an impairment in maternal caregiving behavior, which then might result in a growth retardation phenotype and a lower survival rate. So in our view, adding ultrasonic vocalizations on top of the other behavioral assays we do can tell us a lot in addition. That's why we really think that it's a good idea to add those vocalization recordings in animal models for mental disorders. Bernhard Engels just mentioned SHANK3, the SHANK3 model for autism, but of course there's many others. I gave an example here, bipolar disorder. So our feeling is that in general, it makes a lot of sense to include those vocalizations in the behavioral phenotyping battery, and that we really need to listen to our animal models literally. With that, I want to thank the funding agencies, particularly the German DFG and the Flemish FWO. I want to thank my mentors, Jackie Corley, Francesco D'Amato, Thomas Süthoff, and Rainer Schwarting. Of course, I want to thank all the collaborators. I mentioned Markus Wöhr, I mentioned Paul Clark, for example, but there's many, many more. And of course, I want to thank the people in the lab who actually do the work. And particularly, I want to thank Tianhua Wang, who did the TPH2 studies that I presented today. So she's a very hardworking and very talented PhD student in the lab with already 3 publications. So I'm very happy about that. So really nice work. And of course, I want to thank you for your attention and want to close with one quick comment about our special issue on ultrasonic communication in rodents in behavioral brain research. We do have right now already about 15 to 20 articles accepted, which are already accessible online. So you can look them up if you're interested. From many, many labs also represented here at this conference. So thank you very much for your attention.